I'm happy to be here, and I want to tell you something about evolution-guided sequence landscapes and protein design. So in principle, it's quite different from what we had in the morning today, but there are some words like sequence, like landscapes or so, which actually will have a very similar meaning to merci beaucoup. No. Which will have a very similar meaning to what, for instance, Eric was talking about in this morning. So it's a project which was done with some people here in Paris, some students, some colleagues, and together with two experimentalists from the US, which are Bill Russ and Rama Ranganathan. Okay, so the starting point is, so I'm also like the speakers before by training a theoretical physicist and got somehow into biology, and my way into biology was actually via data, via inference, so one of the important points in biology is now that there's some quite important transformation towards what you could call a data-rich science, and it's represented. Do I have? This is a pointer. Yes. So it's represented here by one database. Which is, so it's about protein sequences, because I'm intended to talk about proteins. And it's the Uniport database. And it's basically a database where all sequence proteins, all protein sequences are deposited. But there are some parts, there's one part which is just amino acid sequences. So it's basically, if you have a genome sequencing project finished, you can bioinformatically predict sequences of proteins from the nucleotide sequence of the sequence genome, and then they are put into this database. So basically, you just know the sequence and nothing else about these ones. And you see this number has had quite some increase in the last years, in particular thanks to high throughput sequencing. And it's now slightly more than 100 million proteins. On the other hand, there's a sub-database, which is a so-called SwissProt database, which contains all those proteins which actually have human annotations. So somebody has been working on this specific protein, has found out something about this protein. Can be functional annotations, it can be a protein structure, it can be various information. So it's not necessarily a homogeneous information, but there's some information which was put into this database. And you see this number is much smaller, and actually it stays almost constant, has been almost constant over the last almost 10 years. And which does not mean that the knowledge about these proteins is constant. So the annotations are updated all the time, but it's basically the same protein, because it's proteins coming from model organisms which are studied a lot and so on. And that's why basically you have better and better knowledge about a small set of proteins as compared to this huge number of proteins where you just have sequence information. So there is some enormous gap. And with these low throughput experimental techniques, obviously you cannot overcome this gap. And so it's a question, how much can we do with computation, modeling, which can somehow in an easy way complement the high throughput experiments. What's actually helping, and that's evolution guided in the title of my talk, is that these protein sequences can be organized into so-called protein families. And so the idea, a protein family, is a set of proteins which have some common evolutionary ancestry. So basically, so they are so-called homologs. So basically, these are the same proteins in different organisms or in different pathways. And these proteins typically have a quite conserved biological structure and function, because they are doing sometimes basically the same thing, using the same mechanisms, but in different species. But if you look to the sequence level, actually, they are very, very strongly diverged. So typically, in large protein families, you have something like 20–30% of the amino acids are identical between two sequences, and the other 70–80% are exchanged, are mutated in the course of evolution. So you have a lot of variability here. But if you take any of these individual proteins, and for instance, you introduce a small percentage of random mutation, it's very likely that you destroy this protein. So actually, the strong variability must somehow have traces of what is here conserved in the structure and function. So here, for instance, I just have an example. These are two proteins which have, in the displayed region, they have about 30% sequence identity. And you see that the structures overlay perfectly. So they are almost identical in structure. And okay, and what is nice about these protein families, there are big databases like the PFAM database, which provide these as multiple sequence alignments. So the data I actually have are data which look a bit like this one, are big matrices of aligned sequences. So typically you have something, so each line here is one protein. You have many proteins which are aligned. Typically, they have something like 50 to 500 positions. And you typically have, for good families, you have something between 1,000 and 100,000 sequences in this alignment. Sometimes it may, in extreme cases, may go up to 1 million. But in some other extreme cases, maybe just be a handful. But I cannot do anything with those ones. So I want to talk about things which have certain abundance. And now my question is, what can I do with this kind of table? What can I learn from this kind of alignment? What is actually, how much does the variability in these sequences tell me about the evolutionary constraints which were acting onto these proteins and which are this conserved structure, the conserved functionality? How much can I get out of this kind of data? And if you look to this table, there's something which is quite obvious. For instance, here is a column which is all H. And actually, this protein is called histidine kinase. So it has to be a histidine here. Whereas I have highlighted another column which looks wildly mixed. So it uses a lot of distinct amino acids in this column. And so that's obviously, you see, it's not just a random matrix. So there is some structure in there. How can we get something out of this and can capture the variability of the sequences in what I want to call a sequence landscape? And so when Hervé asked me to give this talk, he asked me I should in the best case have something like a quarter or a third of my talk on the blackboard. And so I decided to explain a bit how this modeling can be done here. So what we would like to do, we have this multiple sequence alignment. So multiple sequence alignments will always look like this on my blackboard. MSA, multiple sequence alignment. And I would like to capture the sequence variability. So how do I want to do this? I want to write something which is a probability distribution of a sequence. So A1, AL. And these AI here, AI are amino acids. So let's call this A, which is A, C, I'll call it like this, A, C, and so on, the amino acids. Or as you can see there, it can be a gap if there is nothing which is alignable, which corresponds typically to a deletion in the sequence. Okay, you have, so I would like to write something like this. And so the relations to landscapes in the sense of Eric van Nimwegen is clear because I can write the log P, this will be the landscape. So it's exactly the same concept. But if you look to some very simple numbers, you will see that this is not a trivial task because I told you that we have something like m 10 to the 3 to 10 to the 5 sequences. Whereas here, this one is 21 to the L numbers in principle, because I have 20 amino acids and this gap, which can be in the sequence. And so L is something like 50 to 500. So this number is enormous as compared to what we have in data. So we cannot do this. So we cannot do what we do with coin tossing, that we just toss and that we just count the number of heads and tails and get probabilities from this. We are always extremely undersampled. So even if you look to, so this would be all sequences, which doesn't make much sense to talk about this. But even if you, there are some estimates about functional sequences. I'm not sure how reliable these estimates are, but they are still something like 10 to the 0.4 L, so which is basically 2.5 amino acids possible per position out of the instead of 21. So this is a huge number, and so we have to somehow reduce. We have to find some modeling approach which still allows us to estimate this one for this year. And one idea to do this, which was already mentioned, is maximum entropy models. And what do we do in the maximum entropy models? First, you have to select a certain number of observables. And what is an observable? An observable is O mu of, it's basically a function which goes from an amino acid sequence, so some A to the L, to real numbers. So it's something which you can observe on the sequence. Can be very general. It can be, for instance, the fraction of alanines, or it can be, I don't know, the hydrophobicity, or you will see what I will use. So, but anyway, it's something which you can easily measure on a sequence. So now you can, and we choose a certain number, mu equal 1 to p of these observables. And now what we would like to have is that actually our these observables have a certain statistics on the data. So in particular, we can calculate the average of these observables O mu over the data, which is basically just the we measure on each sequence, and we average this over all the sequences. And what we ask for is that our model here is coherent with this, so that our model reproduces this average. So this would be that we have a sum over all possible sequence P of A O mu of A. So this is data average, and this is a model average. So that's fair, that our model reproduces whatever we decided are important observables in our data. So now, having this, we would like, for some reasons, we are convinced about a certain number of observables. And I will talk about how and to select and so on in a moment. But if we are think that a certain number of observables are actually describing our data, then we would expect that this probability distribution, which we want to construct, should be constrained only by these ones and by nothing else. So subject to this constraint here, it should have maximum entropy. So this means we have seen the equation before. Log P of A should be maximized. OK, so this is a very simple optimization task. We have to optimize this under these constraints. So we introduce multipliers. So we do something like this: P of B of minus sum over A, P of A log P of A plus sum mu equal 1 to P sum over the A. We have some lambda mu times, let's write it like this, lambda mu sum over A, P of A, O mu of A minus this data given this one plus some normalization constraint. So it's another constraint which just says that the sum of the P of A is equal to 1. And if you perform this, you easily find that the form of the P of A is a form which resembles very much a Boltzmann distribution. It's an exponential model which is written in this way: sum mu equal 1 to P lambda mu O mu of A. Okay, so you find a very simple analytical form. Obviously, you have to optimize these ones actually to meet the constraints. So this is an analytical form. And this is what I call minus the landscape. And I will also call it minus H of A, because this looks pretty much like a Boltzmann distribution. So now, if you look to this one, you see that I basically didn't tell you anything real, because this one depends on the observables. I didn't tell you what reasonable observables are. And actually, this method in its own doesn't tell you anything about observables. So the question is, what should we use as observables? And actually, that's an important question. I don't have a very good answer beyond taking your intuition or taking prior knowledge. So, but this is something putting into the data before. But there is one point which I think is quite interesting, is that you could verify still if your selection of the observables was a good one, in the sense that it was sufficient to describe the variability of your data, by just taking other observables which are not in here, which you decided not to take in your modeling, and look if on these ones also the model reproduces the statistics of the data. So you use something which you have not fitted, and you ask your model to predict what it does. You can check on the data if it does the same thing. If it does not the same thing, you know that your model has missed something. So you can test, use other observables not fitted, check consistency model data. And if this is actually the case, you would say that your observables define something which is called a sufficient statistics. Okay. So that's the general idea, but now where do we take these observables from? And the first thing is actually what I told you in this alignment up there. You saw that there are some columns which basically take always the same amino acid. So here I take an arbitrary letter, and others take distinct letters, A, B, C, maybe the A turns, the B, and then the D. Something like this. So you have different patterns, and these can be captured by the frequency of amino acids in a column. So what is the fraction of sequences which actually have amino acid I in this column I here, where this one's from 1 to L. And it's just 1 over m sum over m equal 1 to m delta A I m, where I should write this. These sequences here are sequences AM with m running from 1 to m. And if you look, this is actually an average over my data, and this one is my observable. And what is the observable? The observable is just the question, is there amino acid A in position I? So the answer is 0, 1. So it's a very simple observable, but it's an observable because it gives me a number for a sequence. So if I actually plug this into this maximum entropy framework, I get something which is typically called a profile model. Or which is equivalent to what Eric mentioned as weight matrices. And in this case, the probability of a sequence is just an additive function of single terms, so sum over I hi of ai. So it's just, so it is a model where actually you assume that each position is independent from each other, but each position can be biased because, for instance, this H may be very big in histidine for the first column and maybe basically 0 for the second column. And what is important is here you have this I, which is so you have some position specific. So positions are independent, but still your model is position specific. So this is one, this modeling framework with profile models is one of the most successful modeling framework in bioinformatics, because it is at the basis for multiple sequence alignment. It's at the basis for homology detection. And I told you that my alignment there are homologous proteins. So the construction of this alignment in both cases here uses this kind of modeling framework. It is actually at the basis of genome annotation via detecting homology. And since I told you that homologous protein families have conserved structural and functional properties, if you find homology, you can, in a new genome, predict structure and function for what is homologous to something you know already. So it's at the basis of structure prediction, protein structure prediction, in particular homology modeling. Okay, so it's extremely nice, but it's a wrong model. There was, some of you may know this citation, a statistician, George Box, I think he is the name, said that all models, and it was about statistical models, all models are wrong, but some are useful. And that's one of the best cases I know for this statement. It's an extremely useful model. So all computational biology would be unimaginable without this model. Still, it's wrong. And why is it wrong? It's wrong exactly in the sense which I said here. It does not reproduce some of the things which you can observe in sequences, which actually are important in sequences. And one of these is the covariation of residues. So in a cartoon picture, you have your protein, which will fold. And when it folds, it will bring together residues which are quite distant on the chain. And to bring them together, so they will have some A, B here. And then if you bring them together into contact, they physically interact. And not all pairs of amino acids are favorable to interact. For instance, you could have two like charges, and so they would be repulsive. So not all combinations here actually of amino acids are equally valid. And typically, if you make a mutation in one, this will have a slightly deleterious effect on the protein. And you may compensate by the mutation in the second one. So you will observe a correlated usage of amino acids. So maybe you will have here a C, D, then you will have another A, but this comes together with B, A, B, C, D, E, F. But in this case, these two are really not independent. So you will find something as some non-trivial observable, which is a co-usage, co-occurrence of amino acids, which is a character which is given by this quantity. It's a fraction of sequences having A, B together. So if we take just the A, B here, we have 1/2 of the sequences having this one. And so this can be again written M equal 1 to M delta A, AI, M delta B, AJ, M. So it's a number of, it's a fraction of sequences which have amino acid A in position I and amino acid B in position J. This fraction, and this is typically, and again, so this one would be the corresponding observable, which is like this one, but just asking the same question simultaneously for two positions. If you look to data, you have these two quantities are correlated, so it does not factorize in the two single-site distributions, but in this profile model, it would do. So the profile model is not a perfect model to capture the sequence variability, to capture all of the sequence variability. It captures important sequence variability because all these methods work fantastically well, but it misses part of. So now if you take also this into account, you find a model which looks like P of A, some normalization factor plus. Now you have these pairwise terms which lead to pairwise couplings, Jij, Ai, Aj, plus sum over I, Hi of Ai. And these ones here are called direct couplings. So these are statistical couplings. And the method, the whole method is called DCA for direct coupling analysis. What you see here looks a bit like a Potts model in statistical physics, or like a Markov random field, or a graphical model or so in statistics. So there are many names for the same object in different communities. And what is important is you have these dependencies between different sites. So this is a coupling between residue I position and residue position J. And this is actually computationally putting a real problem, because here this is just the same thing like product over I, Fi of Ai. So once you have measured these observables, these hi are just the logarithms of the fi's. So it's very simple to get these parameters. Here, it's complicated. It's an NP-hard problem, so there is no method which is proven to be polynomial time or so all the time. So there is no efficient way. There are a lot of approximations which you can use. There are you can have fast methods which are crude approximations. You can have very slow but detailed methods which get you very good approximations. And I will more talk about the other ones. I don't want to go into the detail how this is done. If somebody is interested, I'm obviously available to tell about this. But what you find is basically more or less, if you look to the strengths of these couplings measured by some norm, and you make a histogram, and I make a log of counts, you find typically something which looks a bit idealized like this. Typically, you don't find so clearly this that you have, and since this is a log, this is really a difference. So you have here a sparse network of strong couplings. And these are residue-residue contacts. I will show you some results in a moment. And here you have a dense part of small couplings. These couplings are important to actually achieve the task to reproduce these observables. We cannot do without these. But the interpretation is one of the big open questions. And as we don't know what these are really, what they really represent, and probably it's a very stupid representation of them, probably you can do much better. OK, so that's more or less what I wanted to do on the board. Are there any questions about this kind of derivation of the model?
So you mean in the same columns, obviously they are not independent, so that's the problem. Why we should introduce the...
Yeah, if you have a here, you always have a B here in this very simple. So typically the patterns are a bit more complicated. But the point is, it's exactly like in the last talk. You have some mutual information. So if you know the state of this one, you get information about this one. So they are not independent, which is said simply by this frequency being not a product of the individual frequencies. And if you look to the data, really there is strong correlations in there. And this is not reproduced by the profile model, but it is obviously reproduced by this model because I can fit all of them. So it's not a miracle that I just fit them. So I should drink this coffee. Yeah, it's quite disgusting.
So you here on this last plot you distinguish small and strong coupling. So
yes.
It has to be small or large compared to something. So what are you comparing to?
This is a very so if you have a very large protein. So in a small protein, you don't see really this. But in a very large protein, you really see some almost a kink. You see that there's some distribution of the bulk, and then you have some tail. And so this really looks like two distinct distributions, where really the cutoff is not. But in principle, if actually you take a protein structure, so you know who's contact and who's non-contacting, then you find that the contacts have this distribution and the non-contacts have this distribution of couplings. So you can, since in a known protein structure, you can distinguish between contacts and non-contacts. You can make this. And actually, this one is completely dominated by contacts, and this one is completely dominated by non-contacts. And what you call small or strong is basically given by the intersection of these two. So it's nothing. So it takes some quite universal values if you look to different protein families. So there is some specific. It depends on the approximation scheme, but within the same mathematical approach to infer these parameters, you get for different protein families the same value. For instance, if you look, if you do a pseudo likelihood maximization, this one here is about 0.3, 0.25, 0.3. So you find a point up to which you have basically only contacts, and then it goes down.
Does the amount of data, so the amount of sequences that you start with, put some threshold on this small?
Yes. Yes. Let's say the point is if you have less data by noise, this one gets broader or this one gets smaller. So they are less clearly separated. Still, the contacts, the most likely coupling between two contacts is very weak. So there are some very strong, but the most likely is still weak. So you find a small percentage, a certain percentage of contacts. So to have this picture, you should have a good alignment. So typically, that's why I insist on this 1,000, which is some kind of an empirical threshold that you have to have roughly 1,000 sequences. If you have much less, basically, this dense weak part becomes bigger than the sparse strong part.
I was wondering, because here, like, you're really going to depend on the metric, how you see the sequence, its position along the amino acids. Sequences. So if I can take the example of the loop here, you could imagine that the length of the loop is going to vary a lot.
Yes.
So like in the positioning of the amino acids, you could have a large variability in the distance.
Yes, but that's in the alignment basically taken into account by this internal gaps. So most of these internal gaps, for instance, here you see that there's a region where you have a lot of gaps. So this is very likely a loop, and it is a loop. Whereas here in this region, you have no gaps at all. And this is a well-aligned helix. So this is a 2-helix protein, which has 1 helix, loop, helix, return.
But you said that the alignment was done on just the local probabilities, how you can find this kind of loop from
So the alignment is done just to maximize under this kind of model. So you try to, it's basically anchored in positions like this one, which are very conserved. If you have a few positions like this one, or here you have this P, which is almost conserved, which is a proline which introduces a kink into a helix, and so on. So in sequence alignment, you try to find conserved sites. And these conserved sites, 8 are some kind of anchor points. And then you have some secondary information, which is, for instance, hydrophobicity, hydrophilicity, because you would like to align the core to the core and so on, which helps you. So there are several levels of, because amino acids are, the similarity of amino acids is taken into account. But so we assume that this alignment got given and correct, which is not completely true, but which is sufficiently true if you have a large alignment. The noise is obviously less strong as a signal.
So you say, for example, that the alignment is wrong.
If the alignment is wrong, it can give you something completely wrong. For instance, if you take to some structures which are hard to align, like coiled coils, if you apply this, you sometimes get coiled coils are very rigid, but sometimes you get the signals that you have a very strong coupling between here and here. They can never get together, but it's just an issue that you didn't align well. So it can create, but over these 17,000 protein families in PFAM, if you are beyond a certain coupling, you have a probability of 0.99 to be a contact in known protein structures. Huh?
Can you use the direct coupling analysis to realign the protein itself?
That's what we try to do at the moment, but it's hard. It's hard to actually get something which looks significantly better than which is done by these profile models.
Excuse me. I get a bit confused about the functional form of H and J's. Are they linear?
So the H's are just, it's a 21-dimensional vector. So it's one H for each amino acid, or the gap.
It's a linear function, Hi times A?
No, it's so you cannot multiply times A because A is A, C, D, and so on. It's amino acids. It's categorical variables. So it's categorical variables. That's why it's really a function. It's just a 21. There's 21 numbers, and this is 21 times 21 numbers. So it's a matrix of interactions.
Just another question. You plotted here the log of count over the absolute value of J. If you would plot the average value of J over the distance, would this be a monotonous decreasing function, or would you have some sort of scale in this system?
Okay, so you have a part which is decreasing. So if you average at a certain distance, it's decreasing. But you have very distant pairs which are actually, and here I show you something. For instance, this is one protein, and you see this is one extremity, and this is another extremity, and all these red lines are the strongest. These are the strongest 30 couplings in this protein. And you see you have one which is from the beginning to the end. So you have sometimes a very strong coupling which is very distant. And this is very indicative for having actually that in the folding, these fold back onto each other. So here, in this case, I have plotted, for instance, the first 30 strongest couplings, which are not very close in the sequence. So because I'm not very interested to have a contact inside the helix, because I know there's a helix. There are other tools to do this. So the distant, so these are just distant couplings, the first 30 plotted, and they are all contacts in this case. And you can use this actually for protein structure prediction, just from sequence data. And it's used quite a lot now, this method and kind of sister and brother methods. Or you can use it also for predicting contacts between interacting proteins, where basically this alignment is now not one protein, but contains pairs of interacting protein, and then you look to couplings between the two proteins. And here is one case. This is actually this histidine kinase, which I have shown before. It's a homodimer, so that's why you see 4 helices. But and so this gappy region was in here. And here you see, for instance, the proline. You see the kink. Okay. So it works when you have enough sequences, and it can guide 3D protein structure prediction complex assembly. But that's actually not what I want to talk about most, even if I see that my time is passing. So I want to talk a bit about this part. So first thing is to be able to talk about this part, to test with other observables, we have to do very well this fitting of these quantities. And this is what is shown in this, and that's, as I told you, is computationally quite hard. But as you can see here, in this case, this is for the single one residue frequencies. This is connected to residue correlations, so the part which is the Fij minus Fij. And you see our model. So this is the statistics of natural sequences, and this is estimation from our model by sampling. And you see they fit extremely well. Just remember the name coarse-mate mutase. It works for many protein families, but this one here is one which I will use in a moment even more. So now you could ask, what is with other observables? And here, that's what I still find extremely amazing, and I'm still have problem to believe what I show you here. But we have also, this is tested for several protein families. For instance, this is a 3 residue correlation, which takes the joint occurrence of 3 residues and takes a part which is not easily explainable via pairs. So it's a connected correlation function. And you see they are extremely well reproduced by the model. The model has never seen these observables. It has just seen these ones, the pairwise, but it reproduces the triplets. It is very, very easy to write down a model where you cannot do this. So where you have 3-point correlations which are not at all reproducible by a model which fits the single and pair statistics. So this is something which is actually somehow significant. These plots come over, they look much nicer on my screen. But what you see here, this is a principal component plot. Why do they come? It's a principal component plot of the natural sequences and the sampled sequences. And you see there is some clustered structure which is well reproduced even there. That's again something which we do not fit, but which our model gives us in a sense for free. So there is some indication that actually we don't need to go beyond pairwise couplings, because you could go on with 3 and so on. But I don't see that actually my model does not reproduce. So I don't see the need for 3-point. So is this, are these sufficient? Is my model generative? And what do I mean with a generative model? A generative model means that if I sample from this distribution and I mix the sample with natural sequences and I give it to the most ingenious machine learning scientist, statistician, biologist, and so on, they cannot tell them apart. And one very rigorous way to tell them apart is actually by experiments, so giving them to a biologist and to see what happens. And so I will skip this one slide, but so I will tell you what we did actually with this coarse-made mutase, which is the one I showed to you. So we had this sequence alignment. We do this modeling via the model which I explained to you. And from this modeling, we can do sampling of having artificial sequences. And we have sent these artificial sequences to our biologist friends. And they have done a very nice experiment. So short conclusion, I just wanted to say that these pairwise models seem somehow statistically to capture necessary and sufficient conditions to capture the sequence variability in some limits. And they are actually able to produce non-natural homologs, which in some cases are up to 40 mutations away from the closest natural homolog. So they are really distant, different. It's a 96 amino acid protein, so 40 mutations is a lot. But still, we have a lot of limitations. For instance, we have to use a strong regularization, and we have problems of interpretation. So maybe there is a much better, much more clever way of a more parsimonious modeling, which doesn't use all these. This is 100,000 parameters have these models. It fits functional and phylogenetic correlations. So it does fit stuff which we want to fit and stuff which is just from the point of view of statistics an artifact of the history, how these sequences were generated. And so what we do here is actually we reproduce the functionality of natural proteins. The one question is, can we actually go beyond this and can kind of improve proteins or change functionality using this kind of ideas? Okay, that's it. Thank you.